Hello, hi, welcome here. So we are talking about the story of life on earth, and that's biology. This is Krishnaveni, your biology faculty. So thank you for joining me today. So good evening host, good evening Ashana. So thank you for joining me. So please do like and subscribe my video. So your small like and your small subscription can make a big difference to those who are not even aware of this huge opportunity ahead of them. Fine? Okay. So I guess you people are more interested in learning about the superhero of biology, so am I. So it's good to see you people here with me joining on this Tuesday evening. Okay, so we are here, but today my time is a bit early, so it's at 5 p.m. Okay, so please do like, so don't forget to like, subscribe, and please do let your friends know about it, because so the more it passes on, so the broader the reach will be. Okay, hello everyone, good evening. Okay, so before we go on to talk about today's lecture, so we were talking about the cell on Saturday evening. Okay, so we actually started, we gave an intro to our superhero, so that was a cell. So we were talking about your cell, so what your cell does, why it is called the superhero. I guess it was a pretty short session where we described so much. We just spoke about the cell again and again. We were telling how it actually acts as a superhero, but actually we have not even experienced how it works, because today we are going more into the chapter than on Saturday. So now, here we go into this journey. So we are going to make this journey of biology interesting. For those of you who are scared of biology, don't. So we are here to talk about the story. So it's a natural science where you'll be amazed at how beautifully it has been made. Okay, so we are just studying what is being already made. So that is one good thing. Okay, fine. So here we go forward to this chapter. So before we actually go into today's lecture, so let me tell you something very interesting. So today is 3rd November, right? So we are already into the month of November. So it's been a long year of quarantine. So we have been sitting at home watching online lecture after lecture. Okay, so yeah, it is still tough for us, still challenging. But yes, we are facing it brilliantly. So let's not lose our hope. And here we go forward. So I just want to tell you, 3rd of November is celebrated as National Sandwich Day. So have you ever heard of something like this? It's weird, right? But it's interesting. So there's something called this National Sandwich Day. So if you don't know, so know it right now. So it's called the National Sandwich Day. So it was started in US, okay, so during the time of Great Depression. So they thought everyone who's deprived of food should at least get one meal as a sandwich. So that's how the whole story originated. So till today it is celebrated as the National Sandwich Day. So please go have sandwich, just be happy, okay, so there's no reason for it. So you can be happy as long as you want, because it is up to you. So everything is in your genes, right? So that is what we were talking about yesterday. So yesterday I was talking about genetics, right? So I told you everything is in your genes. So if you want to feel happy, take this as a reason, okay? So celebrate the National Sandwich Day. So it is pretty good, so all of you can have sandwich. So make sure your sandwich is also healthy, so that you don't add junk food into it, but you had healthy stuff, because your healthy lifestyle is more important for your cell. And that is what we're going to talk about, and that is the whole of biology, right? Fine. So going forward, so let me quickly recapitulate what we were talking in the last class, that is on Saturday. So I teach for class 11 on Tuesday, Thursday and Saturday. I teach for class 12, that is your class 12 portion of biology, on the other 3 alternate days, that is Monday, Wednesday and Friday. Okay, fine. Yes, good evening Kriti. Okay, so here we go forward. So what were we talking about in the last class? So we just started with a chapter, that is the cell, the unit of life. So I called it the superhero. So I'm sure now all of you agree with me that it is definitely the superhero of life. So without cell, you and I will not be here, right? Talking. So we will not be discussing biology. If your cell and my cell was not efficient enough, we will not be here, right? Okay, fine. Yes, good evening. Yes, happy sandwich day to you too, okay? Fine. So let's elaborate on what we were discussing. So what is a cell? So we basically spoke about what is a cell, what it is made of, why it is very unique. So I guess you remember why it is very unique, and it is one of the reasons why it makes a living organism differentiate between a non-living one, right? So it is one of the very distinct reasons why a living organism is differentiated from a non-living organism, and that is because of your cell. So your cell is everything in and out, okay? Fine. So then we spoke about the cell theory, where I told you that there were 3 players, right? So who are the 3 players? So we spoke about Schleiden, Swan, and we also spoke about Virchow, who modified the cell theory that we are discussing today. So what actually was told in the cell theory? That all organisms are made of cells and we are products of cells, right? So obviously your body is made of cells, my body is made of cells, all of us are made of cells, right? So, but how does a cell arise? So that is what Rudolph told us. So he told us that cells arise from the pre-existing cells, right? So that is what was the cell theory that we were talking about. So after that, I guess we spoke about the overview of cell. So what did we talk in overview of the cell? We spoke about your nucleus, we spoke about your cytoplasm, we spoke about your plasma membrane, etc., right? So that was just an introduction class. Fine. Now, today we'll be talking more into the prokaryotic world. So how the prokaryotic cell is actually designed, how it is being approached, how it is being architectured. So we'll talk about it very intricately, and then we'll enter into the eukaryotic world. So just like you and me, the eukaryotes are very complex. So talking about the eukaryotic world intricately will take a lot of time, okay? Because they are very complex, they have a lot of members inside them, right? So they're not pretty simple like your prokaryotes with just one cell and saying just the nucleus is present and just the ribosome is present. It's pretty complicated, right? Okay, so one beauty that you can analyze in biology is that every lecture, so everything that we'll talk about, the base, the platform, the milestone for everything is your cell. So without your cell, nothing is possible, right? So yesterday when I was talking about genetics in my live video, if you had seen, we were talking about molecular genetics. I told you the best gift that you can get from your mother and father is simply 2 sets of triplet DNA sequences, right? So nothing more is needed. So that is enough. And your cell will run, your entire life runs on that sequence, right? So leave out all the material problems in that. So we'll talk about biology here. So what runs you and me is your cell, right? It's not something like sandwich or something, but yes, we run a fuel to our daily needs or our daily living is your cell, right? So brilliantly designed, fascinatingly studied. So now we are going to talk about the cell again. So let me quickly recapitulate why the cell is unique again. So if you remember, so we were talking about this slide, it was the same slide. I just got it so that we can again revise as to why the cell is unique. Okay, so the first thing I told you, because of its chemical complexity, your cell is pretty complex. Yes, it's not very simple because it does a lot of functions. It does a lot of work more than you and I could ever think of. So much is the workload on your cell, but still it does it amazingly beautifully, right? So you agree? If your cell does not work, so you are not here and I'm not here. So remember that. Okay, so it has a high degree of chemical complexity and it is also a microscopic cell. Okay, it is also a microscopic organism which you cannot see in your naked eye. So that is also one reason. Okay, and it is a system for extracting, transforming information or energy from the environment, right? So you get energy from your environment, right? So you can't deny it. You can cook, but still the raw material comes from the environment and your body is able to process that information, right? So that is also a reason why your cell is unique. So quickly going to the third point, it has a capacity for precise self-replication and self-assembly. So I mean to say that your cell has faithful copies of itself. It produces faithful copies of itself. So it makes sure that the information is passed on from one generation to another, and thus we organisms sustain, right? So that was the entire concept of evolution, reproduction as well. Okay. So coming into the fourth point, so you have a mechanism to respond to surroundings. You have the ability to respond to your environment, right? So you and I can respond to the changes in weather, changes in the environment. So if there is a forest fire happening nearby, you can sense it, right? You'll definitely make sure you run out of it. At least if you don't save the others, you are sure enough that you will run away, right? So that is our ability to respond. Fine. And then the last point, so your cell is not a single member, it has a lot of family members inside it, and your cell ensures that there is a coordinated function of all of them inside it. Fine? Yes, and I also told you it is a molecular logic of life. So just like you have atoms in physics and chemistry, so the molecular logic of your life is your cell. Fine? So don't forget, if you're alone, so you have 10 power 14 cells with you working to make you happy. So you don't need any other better reason to smile, right? So we were talking about the overview of the cell. So do you remember this diagram that I'm showing? It has eukaryotes and prokaryotes. So we were basically talking about what the cell is made of. It is made of nucleus. Okay, so that is the first thing. So nucleus contains your genetic material. So your genetic material in prokaryotes is not well defined, meaning it is scattered. It does not have a nuclear membrane surrounding it. But in case of prokaryote, it has your nuclear membrane, well-defined nuclear membrane. Along with that, it has organelles, membrane-bound organelles. So that's what we have been talking about. Yes, and both the cells have a fluid matrix which is called a cytoplasm. Fine. So cytoplasm occupies the volume of your cell. And also I told you the difference between cytoplasm and plasma membrane. If you can recapitulate what is the difference between cytoplasm and plasma membrane, quickly tell me. So what is cytoplasm? So cytoplasm is a fluid matrix, but on the other hand, plasma membrane is the delimiting layer of your cell wall. So both are not the same, so don't get confused. They are both distinct terms. Fine? Okay, so I have repeated this. So eukaryotes are made up of membrane-bound organelles, right? Fine. So they are the members of your eukaryotes. So they are the members of your cell. On the other hand, your prokaryotes do not have any membrane-bound organelles, and your ribosomes, so they are one unique organelle. They are non-membrane bound, meaning your ribosome is not surrounded by any membrane, but it's still present in both your prokaryotes and eukaryotes because your ribosome is essential for the production of protein. So the ultimate agenda of why your cell is working so efficiently, so hardworking, is because it has to produce proteins, and that proteins are produced with the help of ribosomes. Fine? Okay. So on the other hand, just like both prokaryotes and eukaryotes have ribosomes, animal cells have another special or unique non-membrane bound organelle, and it is called a centrosome. Fine? So you would have heard about this term centrosome, centromere, and it can be quite confusing. For now, remember what is centrosome. Okay? We will talk about centromere later on. So that is also covered in this chapter. Okay? Stay tuned to know the difference between both. Fine? Okay, so now we were in prokaryotic cells. We were talking about how the prokaryotic cell is designed, or what are the organisms that come under the prokaryotic world, right? So the first thing is your bacteria. So your bacteria comes under the prokaryotic world. Then it's your blue-green algae. Then it's your mycoplasma. So we were talking that your mycoplasma is the smallest organism, right? So do you remember I told you the diameter of mycoplasma? So mycoplasma is pretty smallest, right? So it is 0.3 micrometer, so that is the diameter of the entire cell. So this is the GIF of all your prokaryotic world. Okay, so going forward, we were talking about the prokaryotic cell. So except the mycoplasma, which is the smallest organism, all the other prokaryotes, they do have a cell wall. Okay, so what you have to focus on this chapter is that basically we are going to talk about the organelles. So we are going to talk about the prokaryotic cell separately. We are going to talk about the eukaryotic cell separately. So prokaryotic cell as such do not have much organs. It is pretty simply arranged, simply designed. So we're just going to talk about the modifications of prokaryotes. So how is their nucleus formed, or do they have any special characteristics, like ribosomes are present in prokaryotes. So how is the ribosome designed? That's all. That's all for prokaryotes. But for eukaryotes, you have a lot of membrane-bound organelles that we'll be discussing. So the point that you have to note, or the take-home message that you have to catch, is that you should remember the special features of both the prokaryotic world and the eukaryotic world. Fine? Okay. So the first thing, so remember, except mycoplasma, which is the smallest of all the prokaryotes in the world, they do not have a cell wall. So all the other prokaryotes, they have a cell wall. Fine? So this is the first definition that we are talking about prokaryotes and eukaryotes. So your prokaryotes do not have well-defined nucleus, meaning the membrane, the nucleus is not properly organized. Instead, it is scattered throughout the cytoplasm. Fine. So there are no nuclear membrane. Okay. Then the third point is they have what is called as the extra chromosomal DNA. So these extra chromosomal DNA are called as plasmids. So they are just extra, so they are not their genetic material, but they are present in bacteria naturally. Okay, so they are mostly double-stranded circular ones, so they are called as plasmids. So they are naturally present in your prokaryotic cell. Okay, so that is one of the special features. And today, at this moment, the entire healthcare research and the genetic research is exploiting this extra chromosomal DNA, and that is your plasmids. So plasmids act as a vector. So let me tell you how it acts as a vector. So today we are living in a world where we are talking about gene therapy, gene editing. So if you're well aware, so the the Nobel Prize in physiology was awarded for CRISPR gene editing, right? So we are in a world of gene revolution. So we are in the era of genetics. So we are in the era of genes. Okay, so where everything can be blamed on to genes. So today we are exploiting plasmids to the level that we can. Okay, so what actually happens? So you have heard about designer babies, artificial babies coming up, right? So how it is actually done? So you can take out a gene from a person. Say for example, if you want to take a big celebrity's gene and then you want to put it in someone else. So taking out a gene and putting it in someone else is easier being said than done. Okay, so it is not such an easy process. So it is definitely tedious and tiring to isolate DNA, because if you want to isolate DNA, so once inside the body, your DNA is fine. So when it comes out of the body, it becomes destable. The stability is lost. So it is utmost important to maintain the stability of DNA. So before you transfer it to another person. So how it is being transferred from one person to the other person is with the help of plasmids. So plasmids have a lot of applications that you can talk on endlessly. So the whole day, the whole night will not be enough. But as of now, understand they have extra chromosomal DNA, and that is your plasmids. Fine? Okay. So going forward, so they have no membrane bound organelles, that is their family members are very less. Okay. So basically I mean to say it's a nuclear family. So you can compare it with your nuclear family is your prokaryotic cell, and your big family or the joint family is a eukaryotic cell, because the organelles in eukaryotes are endless. Okay. So they do not have membrane bound organelles except your ribosomes. So ribosomes are present in both the eukaryotic world and the prokaryotic world, which means it's indispensable. It is the essential one for the production of proteins. Fine? Okay. So apart from what is called plasmids, your prokaryotic cell also has what is called as mesosomes. So have you heard of this term before? So mesosomes are the differentiated form of your cell membrane. Okay, so you have infoldings of your plasma membrane which comes inside the cell, and those infoldings are called as mesosomes. So now if you look into the picture, you will understand. So this is the structure of a prokaryotic cell. So it is a nuclear family, so it does not have many members inside your cell. Okay, so it has a cell wall, a cell membrane, right? So if you can look at the figure like this, so that is your flagella. So right now in your body and my body, there are thousands of bacteria floating here and there. So they float with the help of flagella. So that is their locomotory weapon or organ that you can say. It is trying to swim inside our body, and it's trying to ingest or infect us. Fine. So for example, one example I can give you is salmonella. So salmonella resides in your intestine. Okay, so this salmonella can cause your typhoid, it can cause diarrhea, it can cause food poisoning, etc. So the target organ for your salmonella is your stomach, small intestine especially. Okay. So it enters your body through when you have contaminated food or water. Okay, so that's why it's not always advised to eat in an unhygienic place, right? Because you can inject a lot of bacteria and then you suffer from a lot of disease. Okay, so this salmonella enters your body. Okay, so once it enters your body, it has to travel all the way down to the intestine, because its power works in the intestine, because we are also good in only certain areas, right? For example, some people will be good in physiology, some people will be good only in genetics. So your zone of areas, your genre is different. So similarly, this salmonella can reside only in your small intestine. So it will quickly travel, it will quickly swim with the help of this motile organ that it has. So it is called as flagella. Fine. So this is the structure of your prokaryotic cell. So going on further and talking about your cell envelope and its modification. So in the previous figure, we saw that it has a cell wall. It also has a cell envelope. So what are the modifications of the cell envelope? Whether the cell envelope is present as such or it has been modified. So that's what we are going to see now. Fine. So first, your cell envelope has 3 layers. Okay, so this layer, the order in which I'm going to talk is from the outside to the inside. So first one is your glycocalyx. So your glycocalyx is followed by a cell wall, and your cell wall is followed by your plasma membrane. Fine. So these are the 3 layers of your cell envelope. So this is from outside to inside. So you have your glycocalyx followed by your cell wall followed by your plasma membrane. Okay. Fine. So based on the cell wall, based on the design of your cell wall, your prokaryotic cell can be divided into 2 types. One is called as gram-positive. The other one is called as gram-negative. So basically you divide bacteria into 2 categories based on the design of their cell membrane. So in case of gram-positive bacteria, right, so you have a thick peptidoglycan layer. So this is a protein layer that is present on a very thick layer or a thick sheath on the top of your gram-positive bacteria. Fine. Whereas your gram-negative bacteria has a very thin peptidoglycan layer. So this is based on the architecture of your cell wall of your bacteria. So it has been classified into 2 types. One is gram-positive and the other one is gram-negative. Fine. So where do you find the application of this? What is the use of classifying bacteria? Because every day we are not looking at bacteria. Bacteria is looking at you. Do you know? Yes, bacteria is looking at you right now. So bacteria is present on this whiteboard. It's present on this pointer. It's present on this pen. It's present on the screen that you're looking at. Okay. So bacteria is definitely looking at you. But what is the application of gram-positive and gram-negative? So we have a lot of bacterial disease that is affecting humans, right? So you have a lot of bacterial disease affecting dogs as well. Okay. lot of other organisms, even cows have bacterial infection. So if you want to treat a bacteria, you need to kill the bacteria. So your ultimate aim, the pharmaceutical aim, is you have to give a drug so that the bacteria dies. So if the drug has to reach the bacteria, you need to understand the design of cell wall. So what type of drug will penetrate inside the cell wall, right? So you need to differentiate, or you need to study how it is designed. So thus, based on the diagnostic point of view, it has been classified into gram-positive and gram-negative. So it has been done by Hans Gram. So hence, in honor of his name, we have named it as gram-positive and gram-negative bacteria. Fine? Okay. So now going back to a cell envelope modification. So we spoke about glycocalyx. So glycocalyx has two different variants. So just like a bipolar disorder. So you have two phases, right? One phase at home and one phase with your friends. So at home you all behave so decently, but with friends we are completely a different person. That's okay, that's nothing wrong. Okay. So just like that, your glycocalyx has two different variants. The first variant is called as slime. Okay, it is called as the slime layer. So when your glycocalyx layer is present as a loose sheath, it is called as the slime layer. When it is present as a tough capsule, tough and thick, it is called as capsule. So these are the two variants of your glycocalyx. Fine, is this clear? Okay. Talking about your cell wall, so we are done with your glycocalyx. Okay. So talking about your cell wall determines the shape of your cell. Okay. So it determines the shape of your cell. It also contributes to the cell-to-cell communication. So it's easy that your body has thousands of cells, billions of cells, trillions of cells, everything is fine. But how do the coordination has to be maintained, right? How the coordination has to be maintained, that plays a role, right? So even at home, you are 4-5 of you, but still if there's no coordination, you quarrel among each other every day. Is it good, right? It's not healthy, right? So all the cells has to communicate with each other. They have to coordinate. So imagine if your cells of digestive system says, okay, today I'm not digesting, I have to rest. The cells of your heart says, today I'm not pumping blood, let me rest. So imagine what will happen to us. So your digestive system says, I will only digest breakfast today, I'm not digesting lunch or dinner. So then you skip lunch or dinner, kya? You don't do, right? So you have all the 3 meals. Instead you take extra meals. But your digestive system does not question you. So it does not mean that you can take it for granted. So that is why we say a healthy lifestyle is the best one. Okay. So here why I am talking about this context is that there has to be some communication between your cells, and that is maintained by your cell wall. Fine. Okay. Then talking about your plasma membrane. So here PM refers to plasma membrane. So that is, in short, it is selectively permeable. So what do I mean by selectively permeable? So here I mean to say that plasma membrane being the delimiting layer, it allows the movement of molecules into and out of the cell. But at the same time, it does not allow all the molecules to enter and all the molecules to move out. Just like you have your guard at the school gate who allows you only when you have an ID card, right? So when you have your ID card or school uniform, he allows you inside. Otherwise, he does not allow you, right? So similarly, it has certain criteria as to which molecule to accept and which molecule not to accept. Okay. So that is called the plasma membrane. So that process or the event is called as selectively permeable. Fine. So is this clear? So this is the slime layer of your glycocalyx and this is the capsule of your glycocalyx. Fine. Okay. Now going forward, so we'll be talking about flagella, pili and fimbriae. So they are supporting structures for your bacteria. So there are 3 different structures. They are not all the same. So or they are not analogous as well. Okay. So this is your bacterial cell. So you have 3 different structures labeled. So remember flagella, pili and fimbriae, they are not the same ones. So this is your flagella, so which helps in the movement of your bacteria. Okay. So these hair-like structures are called as pili. Okay. So pili basically helps in adherence and it also helps in some transfer of extra chromosomal material like your plasmids. Fine. So pili, they are hair-like outgrowths. They also help in adherence and also in the transfer of your genetic material. Then fimbriae, they are filamentous ones. They also help in the adhesion of your bacteria. So what do I mean by adhesion here? So just like I was talking about the salmonella, which has to reach a small intestine. So once it reaches the small intestine, it has to sit down somewhere, right? It has to attach. It can't be floating because you have all your digestive juices coming out. So it will be washed off. So your bacteria is way smart enough that it comes and adheres onto your epithelial layer of your intestine. Fine. So for such kind of adherence, your pili and fimbriae are essential. Hi Raja. Okay, fine. So bacteria can be both motile and non-motile, no doubt. So when it's motile, it has flagella, and flagella has 3 parts. So it has a filament, it has a hook, and it has a basal body. So these are the parts of your flagella. Fine? Okay. So pili, so we have already spoken about pili. So pili is an elongated tubular structure. It is made up of a special protein. Okay, fine. So fimbriae is another filamentous protein. It looks like a fiber sprouting out from the cell. So it basically helps in your adherence. It helps the bacteria to adhere to the target organ. Okay, so now going on to the next slide, that is your ribosomes. So ribosomes, I told you they are present in both your prokaryotic cell and your eukaryotic cell. The design is almost the same, but with minute differences. Obviously, right? Because eukaryotic cell is so complex, so you need to have a lot of facilities in eukaryotic cell. So just like a joint family is living together, you need a lot of resources in the house, right? You need a lot of rooms, you need a lot of ingredients to cook. Everything has to be in excess. But that's not the case when you live in a nuclear family. So everything can be limited. So similarly, the ribosome of your prokaryotes are also developed, but with some differences from your eukaryotic one. Fine. So let's look into what it says. So your ribosomes are present in the plasma membrane of cells. Okay. So your ribosomes are non-membrane bound. So they are the only organelle that is present in your prokaryotes. Fine. So the size of your ribosome, so it is 50 nanometer by 20 nanometer in size, and there are 2 subunits. One is 50S and one is 30S. So if you ask me what is S, so I have a picture of ribosome. So this is the large subunit and this is the small subunit. So the large subunit here is your 50S and the large subunit here is your 30S. So what does S mean? So S stands for sedimentation coefficient or Schwarzschild unit. So it is the relative or the indirect measure of your density and size. Okay, so it is also a unit which talks about indirectly about the measure of your size and density. So that is why it is called as S. Fine. So the larger subunit is 50S and the smaller subunit is 30S. So this large subunit and the small subunit are not always attached to each other. Only at the time of protein production they come and bind together. So when they come and bind together, they become what is called as the 70S. So separately they are called the 50S and 30S, but together they are called as the 70S ribosomes in prokaryotes. So remember, prokaryotic ribosomes are 70S. Hi Priyanka, good evening. Okay, fine. So this is about ribosomes. One more thing about ribosome is ribosomes attach to a single RNA and it forms what is called as polysomes or polyribosomes. So so let's talk about central dogma. So when I was telling you about central dogma, I said you have your genetic material in DNA, right? And it gets copied on to mRNA. And finally, you have what is called as the proteins produced, right? So the moment your mRNA is produced, your ribosomes will quickly go attach like this, thus forming a polyribosome or a polysome, so that it helps in the production of proteins. Fine? Is this clear? So this is about ribosomes. So going forward, so this is also one of the unique feature of your prokaryotes, that is inclusion bodies. So inclusion bodies are very, very unique and salient feature of your prokaryotes, that is its storage material. So just like we human beings have vaults, you have bank lockers to store all your assets and your valuables. So your bacteria, the only valuable your bacteria can have is food, right? So that is fundamental to them. So they store their food and their resources in what is known as inclusion bodies. Okay. So they are reserve materials present in your prokaryotic cells and they are stored in the cytoplasm and they are called as inclusion bodies. So these inclusion bodies serve as reserve material for your prokaryotes. They get their resources from inclusion bodies and it is not membrane bound, hence it is not called as an organelle. Fine. So examples of your inclusion bodies are your phosphate granules, synophycean granules and your glycogen granules. So these are some of the inclusion bodies present in certain type of prokaryotes. Okay, so phosphate granules, so what do you think is stored there? Basically phosphate, right? So that is what is your inclusion body. So another example, you have what is called as gas vacuoles. They are present in your photosynthetic blue-green bacteria. Okay, that's good Priyanka. So these are your vacuoles. So if you can see here, can you see stack-like structures? So they are your vacuoles. Fine. So now we are done with the prokaryotic world. So we have spoken a lot about the prokaryotic world, if not much. Okay. So what did we talk about? We spoke about the ribosomes. We spoke about the cell envelope modification. So how the cell envelope of your bacteria is modified, right? So we spoke about that. Then we spoke about the ribosome as to how the ribosomes of your prokaryotes are made. And one more important point, we spoke about gram-positive and gram-negative bacteria, right? So next class, give me an example of a gram-positive bacteria and a gram-negative bacteria, okay? So when you Google for gram-positive or gram-negative bacteria, make sure you see the structure of the cell membrane as well. Make sure you see the structure of the cell envelope as well. How it is designed. What is the difference? So if you have a picture clarity, then it stays in your memory forever. Fine? Okay. Now we are entering into the eukaryotic world. So eukaryotic world is so complex. So we are going to talk about a lot about the eukaryotic world. So just like your brain and my brain is complex, so is our cells. So we'll first talk about the cell membrane. So the design of the cell membrane. So the design of the cell membrane itself is pretty complex. Okay. Then we'll talk about the cell wall. So how different your cell wall is in your eukaryotes and prokaryotes. We'll compare both of them. Then we'll talk about your endomembrane system. So when I say this term system, it means it is a group. It has 4 or 5 members associated with it. So that's when you call them as a system, right? That's when you call them as a team. So only when you have 4 or 5 members along with them, or 4 or 5 members in a group, you call them as team. So is the endomembrane system. It is a team consisting of 4 or 5 organelles. Fine. So then we'll talk about mitochondria. So mitochondria is not new to us, right? Okay. So mitochondria is the powerhouse of the cell. So we'll be talking in detail about the structure of mitochondria. Okay, and the functions of mitochondria as well. Then we'll go forward and talk about plastids. So one example of your plastids is your chloroplast. So we'll again talk about the structure of your double membrane chloroplast, how it is designed, how it's intricately architectured so that it can perform the function of photosynthesis. Fine. Then we'll talk about ribosomes. So anyway, we spoke about ribosomes in prokaryotes. We'll elaborate that same thing with respect to eukaryotes as well. Then we'll talk about what is cytoskeleton. So have you heard of this term cytoskeleton? So cytoskeleton is basically a network of filaments and tubules which gives integrity to your cell, okay, which maintains your mechanical integrity of the cell. Then we'll talk about cilia and flagella. You can ask me the doubt, ma'am, we are talking about eukaryotic world, then why are we talking about flagella and cilia? Because just a few slides back we were saying that cilia and flagella are unique to bacteria. Now, if you ask me why are we talking about cilia and flagella here, there is a reason. So, your body and my body also contains cilia. So, we also have what is called as the ciliated epithelial cells. So, do you know where the ciliated epithelial cells are present? They are present in your respiratory tract. So, basically your windpipe. So, they are small hair-like structures like this. So, when you inhale the air from outside, so the air and the atmosphere does not have the same temperature as your body. So it is the function of this ciliated epithelium to first remove the unwanted particle or the foreign particle from your air, okay, and then it will bring it down to the body temperature. So we also have ciliated epithelium cells in our body. Yes, good Priyanka. So that is also another example that is also present in the fallopian tube. Fine? Okay. Then next we'll be talking about centrosome and centrioles. So just a few minutes back I told you don't confuse the term centrosome and centromere. So there's also another term called as centromere, right? So don't confuse both. So they are two distinct terms. Fine. Then we'll talk about nucleus, that is the brain of the cell. So nucleus contains your genetic material. So your genetic material codes for all the informations, right? Now you're smiling, that's because your happy genes are active, right? Now your smiling genes are active, that's why you're smiling, right? So that is nucleus. Then we'll talk about microbodies. So microbodies are small vesicle-like structures or small bag-like structures that stores nutrients, okay? They can also store enzymes, so they are called as your microbodies. Fine? So going forward, let's enter into the eukaryotic world. So what is this? So your protista, so they also come under the category of your eukaryotic world. Then you have your plants, so plants are also eukaryotes. Then you have your animals, they are also eukaryotes. Your mushrooms, your edible mushrooms, they are also eukaryotes. And then last but not the least, you and I, we are also eukaryotes. So we also belong to this eukaryotic world. Fine. Then, so talking about or introducing to you the eukaryotic world. So they have extensive compartmentalization of the cytoplasm. So CP stands for cytoplasm and PM stands for plasma membrane in my slides. Fine. So you have extensive compartmentalization. Just to compare it with your joint family, so you have a lot of rooms. So you have a lot of compartments here because it contains a lot of membrane bound organelles. Okay. Then you have an organized nucleus within the nuclear envelope. So the nucleus has a particular space and it has a proper dress around it. So that is called as a nuclear envelope. Then it also has complex locomotory structures and cytoskeletal structures, right? So we have also cytoskeletal elements, so that is called as your microfilaments and microtubules. Okay. Yes, I am listening to your answers. Okay. So let's go back. It also contains genetic materials, that is your chromosomes. So that is also the reason why a eukaryotic cell is unique. That is why the envelope, the nuclear envelope plays a role here. Then all cells are not identical. I guess you're sure, because all the cells in your body are also not identical. For example, your neurons are not identical to your heart cells, that is your cardiac cells. So everything is different, right? So they are not identical. So plant cells, for example, they have cell wall, plastids and a large central vacuole. Okay. On the other hand, the animal cells have unique centrioles. So is this much clear? So we have just introduced the eukaryotic world for you, and these are the salient features of your eukaryotes. Now, I'm pretty sure the plant cell and animal cell is not a new thing to you, but quickly let's look into the plant cell and animal cell. So can you quickly tell me the difference between plant cell and animal cell? Okay. So yes, quickly tell me. So all those who are listening, Priyanka, quickly tell me the answer. So difference between plant cell and animal cell. So it's not new, right? So you have been reading about plant cell and animal cell from your 9th standard, 10th standard. Now you're preparing for NEET. So this should be one thing that you should be well aware of. So quickly tell me, plant cell, animal cell. So what is the difference? So first difference, okay, so both of them have well-defined nucleus because both of them come under eukaryotic world. So both your plant cell and your animal cell, they have well-defined nucleus. Okay, something else? The most important one, the first one that comes into your mind, it's the cell wall. So your cell wall is present in your plant cell, but at the same time the cell wall is absent in your animal cell, right? Okay, both of them have membrane-bound organelles because both of them are eukaryotes, right? So Priyanka, so that is not the answer, so that is not the difference. Okay? Yes, so cell wall is the difference. Anurag, yes, right. So that is one difference. So cell wall is the difference. Another difference is that we already spoke, your animal cells have centrioles, your plant cells do not have centrioles. But the connection between your plant cell is plasmodesmata, and in case of animal cells, it is desmosomes or your intracellular junctions. Okay? Fine. And one more salient or the important distinction between a plant cell and animal cell is your vacuole. So your vacuole in your plant cell is huge enough, it occupies 95% or 90% of your cell volume. But it's not that your animal cell does not contain vacuole, but your animal cell is not as prominent as that present in your plant cell. Okay? Okay, so you were confused between eukaryotes and prokaryotes. I guess I understood because you were telling the difference between prokaryotes and eukaryotes, but I was asking you the difference between plant cell and animal cell. Yes. Okay, so you say plant cells contain chloroplast. Okay, so that is also right. Okay, so now going forward, let's see. We are going to talk about the cell membrane first. So how the cell membrane of the eukaryotes is distributed, how it is designed. So it is mainly composed of lipids and proteins, right? So we are talking about the design of your cell membrane. So come back, we are talking about the eukaryotic world. So we are talking about the cell membrane specially. So it's mainly composed of lipids and proteins. So major lipids or phospholipids, and they are present in 2 layers. So it is called as bilayer. Fine. Then it has, okay, so let me show you the picture first. So this is how your cell membrane is designed. So look, it has 2 layers and it is made up of lipids. So it is called as phospholipid bilayer. It is basically made up of lipids. So it is phospholipid bilayer. Fine. But how is this arranged? If you can see the round ones here, and then it is followed by a tail, right? excuse me. So this round one is called the polar heads, okay? So they are called the head, and the tailed one is called the non-polar tail. So your head one is hydrophilic and your tail ones are hydrophobic. So what do you mean by hydrophobic and hydrophilic? So hydrophobic is water fearing, hydrophilic is water loving. So your head ones which are polar, they are hydrophilic, okay, so they love water. Your tail one which is non-polar, they are hydrophobic. So this is how it is arranged. Now if you look, this is again a polar tail and a polar head and a non-polar tail, right? So this is how they are arranged. So that's what is written on the slide. So hydrophobic tails are towards the inner part, right? And the non-polar tails of saturated hydrocarbons protect from the aqueous environment. Yes, Priyanka, that's right, okay? Apart from your phospholipid membrane, it also contains cholesterol, okay? Fine. Talking further, apart from lipids, it also possesses proteins and carbohydrates, okay? So the ratio of proteins and lipids, they vary considerably. Just like all the cells in your body are not identical, the proportion of proteins and lipids are also not identical. They vary considerably with your cell. Hydrophobic means not water loving, phobic means fear. So hydrophobic is water fearing. So philic is loving, okay? So I think you have interchanged, Anurag. So hydrophobic is water fearing, hydrophilic is water loving, okay? Okay, so you said it as water hating, okay, fine. But just change it, hydrophilic is water loving, hydrophobic is scared of water, fine. So for example, so I just want to give you one example where the proportion is considerably different. So one such example is your erythrocytes. So your erythrocytes are basically your RBCs. Okay, so your RBC is 52% of protein and 40% of lipids. So you can see the difference, right? So apart from lipids, you have two types of proteins. It is called as integral protein. Okay, so let me rub. I have written on top. So you have two types of proteins, integral protein and membrane protein. Integral and peripheral. So integral is also known as membrane protein. So from the literal term, peripheral means the one which is present on the outside. Integral protein is also known as the transmembrane protein. So basically these are your, so this is your integral membrane protein, they transverse through your membrane, right? So peripheral protein, it is just present on the periphery, okay? Fine. So next we will talk about the fluid mosaic model. So fluid mosaic model brings us to the topic of plasma membrane. So what did I tell you about plasma membrane? So it is a delimiting layer present in your cell, right? So what actually your plasma membrane does? It actually enables the movement of molecules into and out of the cell. So based on this observation, they came up with a theory called the fluid mosaic model. So it was proposed by two heroes. So they were called Singer and Nicholson. So they introduced what is called as a fluid mosaic model. So based on this theory, it was said that the quasi-fluid nature of lipid molecules, okay, enables the lateral movement of proteins within the lipid bilayer. Okay, it's not Sanger, it is Singer and Nicholson. Okay, so it is this quasi-fluid nature of lipid. So your cell membrane is made up of phospholipids, right? Your plasma membrane is made up of plasmolipids. So it is because of this lipid nature or the quasi-fluid nature of lipids, that is, there is lateral movement of molecules. Only because of there is lateral movement of molecules, your cell is able to respond to the environment. It is able to communicate with the other cells. So this was one of the defining or the most definite finding or the commendable or the breakthrough finding that came into the discovery along with the cell. Fine. Okay, so the ability to move within the plasma membrane was later referred to as the measure of fluidity. So that is why it allows movement of molecules into and out of the cell. That's fine, okay? So this fluid nature of your plasma membrane is important for various reasons. So the first thing, it is important for your cell growth, right? Then for formation of your intracellular junctions, so the basically connections between your animal cells, okay? Then for secretions, then endocytosis and cell division. So this is how it is, the fluid mosaic model. So it enables the lateral movement of molecules into and out of the cell. Fine? Okay. So moving on further. So we'll stop here today with this much because I think it's getting saturated. So the more we talk about prokaryotes, the eukaryotes, it is never ending. So even if you give me all day, without a break, we can talk about prokaryotes because there is so much. But a human brain can actually concentrate only for 15 minutes, right? So that is a known fact. So already you are saturated with more of classes online. So let's make it a very short one. So I guess I have taught you enough concepts for today. But do join me on Wednesday, on Thursday. So we'll continue with cell, the superhero of biology. So we are yet to talk a lot about the cell, so especially the eukaryotic ones. So we'll talk more in detail about each of the membrane-bound organelles of your eukaryotes, and we will meet shortly. But before that, make sure you like, you subscribe, okay, and be there for my next class. Especially tomorrow I'll be talking about genetics, so be there for genetics. So the 3 days in a week is for class 11 and 3 days in a week it's for class 12, but make sure you join me on all days. So it is a revision, so though you're studying class 11, fine, but make sure you also listen to class 12. It's not going to go waste, right? So join me, and I promise I won't disappoint you. And thank you for joining me now, and have a nice evening. Bye. Thank you.